but without them in this position of knowing a little bit about biodiversity. Now, in the last 250 years, maybe we got to describe through this meticulous and tedious work, maybe 2 million species. And we saw in one of the plenary presentations that we maybe estimate that we have 10 million species, depending on if we include bacteria or not, and so on. So we know only a fraction of the biodiversity out there. And from that fraction, we know even less about those species beyond species names and descriptions. So DNA came to help us about 2 decades ago or a little bit more. And it brought a revolution into how we look at biodiversity. And I'm going to use the term DNA technologies to be a bit more inclusive. So it is DNA barcoding, metabarcoding, environmental DNA, qPCR, microsatellites, RFLP, whatever you are using to look at biodiversity and to look at the 3 levels of biodiversity. So in these 2 decades, we arrived at a massive amount of DNA sequences that are being clustered through algorithms. We've heard about this for the past 2 days. We have currently more than 20 million DNA sequences just for multicellular organisms and much more for bacteria, fungi, and so on. But we have species names for a small fraction of this. So this is where we are today, and that's why I thought this would be an interesting discussion on where we are going in these conditions that we have today, that we will not stop technological innovation and we will not stop the accumulation of DNA data. So where are we going from here? And for this, I have here a panel of Fantastic Four that I will be introducing one after the other, and then I will invite to take a seat here. First of all, we have Regina Wetzer, who is actually a taxonomist. Maybe it's among the very few taxonomists at this conference. So she actually has experience in naming and describing species of marine isopods. For those of you who do not know what isopods are, they are distant cousins of shrimps. They are really cool. She is a curator. She works in a museum. She's a curator at Natural History Museum in Los Angeles, and she's also the director of the Marine Biodiversity Center there. Regina, please. As we probably know in this room, she is a representative of an endangered group, taxonomists, but we are happy to have her here. We are moving on to Evgeny Zakharov, who is not really a taxonomist, but he started in a taxonomic lab. He worked on Lepidoptera, so butterflies and moths. And then he started accumulating or incorporating DNA sequences in his work. He got interested in this, and now he's the director of the Canadian Center for DNA Barcoding. This is the largest molecular lab in the world that is producing DNA barcodes for multicellular organisms. So he is ultimately responsible for the quality of the molecular data that goes into the barcode of life data system. No pressure, Evgeny. And next we have Tobias Froslev. He also started a little bit looking at molecular characters. He's not really a taxonomist, but that's where he started. He says he's a taxonomist. Then he also became interested in DNA, and he started incorporating DNA into his work. And now he works for GBIF. He's a science officer at GBIF. In case you don't know what GBIF is, they have a booth downstairs. You are welcome to go and learn more about it. Tobias. And lastly, but not the last, we have Maylene Gonzalez. She's not a taxonomist. She's a molecular ecologist. So she is using DNA sequences to look at communities. She's also interested in fighting a little bit illegal wildlife trade in Colombia. She works at the Humboldt Institute Humboldt Institute is one of the organizers of this conference, and we are very happy to have Mylene here. Now, I do have the questions written down because I'm a bit stressed about time. And I will ask each one of you, starting with Regina and Evgenia and Tobias and Mylene, to answer a question. Since you are all familiar with biodiversity inventories and also with the use of DNA in biodiversity inventories, from your point of view, what is the most exciting and the most concerning aspect of how we are using DNA technologies in biodiversity inventories and how DNA is reshaping what we know about biodiversity? And I'll just pass the microphone to each one.
Thank you very much. So I'm at the second largest natural history museum in the United States, and I work on a group that's quite esoteric that many think is marine isopods, roly-polies. I get paid for it. I'm at an institution where my director is a dinosaur specialist, so the outreach from a public perspective is about the big charismatic things. So there's always a major effort of making the little things that fill the vast collection spaces, make them available, especially when they've been preserved in various ways, either pinned or put in alcohol bottles, and they're no longer as beautiful as, you know, a nice furry panda bear or something like that. So the exciting part about these inventories, and I use DNA as well. I have been trained as a morphological taxonomist, but as many of you know, if you are from the United States, you can't make a living at it, and I don't really put that on my resume at home. When I took my PhD mid-career, I was definitely discouraged from it and hence moved to molecular science as well. So I use a combination of DNA barcoding and other kinds of DNA resources in order to more quickly and more effectively, efficiently identify new species and biodiversity. So I use existing collections and type material, but again, when I'm looking at new species descriptions, I move ahead with DNA oftentimes faster to sequence everything and then look for patterns and then go back and do the morphological work that needs to be done. But translating that also to the public is a major engagement that is a necessary component of being a museum member. So it should be enough.
All right, thank you. So I'll correct Adriana that I'm not responsible. I'm responsible for the quality of sequence data, yet I'm not responsible for the quality of the collateral information that our collaborators submit alongside those specimens. So this is where a lot of problems come from. But for me, as a young scientist, as Adriana mentioned, starting my career in the hardcore taxonomist lab of entomology, who wasn't prepared to spend hours dissecting genitalia and counting hair on the body of a fly, I saw the technology as the promise of a bright future that now DNA will solve all the problems. Young and naive I was. Yet I am beyond impressed how far the technology has gone, allowing us to cut the cost and accelerate the rate and the volume of these bioinventories and growing these massive data sets. This is all amazing, and it actually opens so many opportunities. But I'm also concerned because I personally contribute to part of the problem with data volume exploding, but the taxonomic curation and our capacity to catch up and validate those records is lagging behind, and that impacts our ability to really go and apply this information in any significant context, like whether it's biodiversity assessment, impact on remediation. So this is why I would like to see more progress.
Yes, thank you. Yeah, just catching up on the taxonomy thing. I started as a mushroom taxonomist and discovered the DNA tool. So I've sequenced thousands of types and also described many mushroom species, but that's what sort of brought my life into DNA, were the starting point of taxonomy. So I still think that's super fascinating and is continued to be a good way of applying those things. But my focus is more on eDNA technologies these days. And what's fascinating about this is that we can now detect and describe biodiversity much more comprehensively than earlier on. We can detect those 95% of the tree of life that we cannot detect with our eyes or anything. So I think that's super interesting. What's concerning, I think many believe that these DNA methods is a threat to classical taxonomy. I actually think it emphasizes the need for classical taxonomy. So I'm overly optimistic at that point. I'm more concerned about people who don't know or who don't understand these techniques and their limitations will put too much emphasis on all the wrong detections that sort of in event that do come with such methods. So I think we need to be very pedagogical in the way we communicate it to people who want to apply it to policy making and things like that. Mm-hmm.
I don't know why mine is the one making songs. But well, for me, I think like coming from ecology and now working in a conservation field, I will say that DNA barcoding and, for instance, eDNA opens you a new field of opportunities and questions, and you can understand, I don't know, like address the question about the diet of a species, or to visibilize, for instance, microorganisms, bacteria and fungi, by like in a context of conservation. No, so in environmental licensing, environmental impact, what is the impact of microbes? So far, microbiologists have been working with their group, but this is not, for instance, a group that has been really visibilized for ecologists in this context of conservation because it was too hard to study. Soil biota is another group. Zooplankton, you know, like things that are super challenging from the taxonomy perspective, and that now DNA just like allows you to at least address those questions. So that is what is exciting for us. And what is like concerning or challenging is that we do understand that there is this interlink between the taxonomy, the DNA reference library, for instance, and the application then of eDNA. But sometimes you have the feeling that either taxonomy by itself and reference libraries are being like forgotten, no? Left behind. And you make most of sense of all the eDNA when all of these other two components are being enriched. So I think the concern will be that recognize that the paces of information are different, but that the three of them are important and that we continue to work in the three of them. Because species with names and its natural history behind are more tangible than numbers. And that I think that will always be.
Thank you so much. We already have a lot of information, and I think everybody is concerned or is excited about similar things and concerned about similar things. So we have this massive amount of data that does not have names or descriptions. And we need to decide as a community how we are moving forward and where we are moving forward. But let's start a little bit, step by step, and we will start with taxonomy, of course. Regina, do you think we can still speak about species in the traditional sense when we have this massive amount of molecular OTUs that are unnamed? They have identifiers, but they are unnamed, and they are being accumulated at the speed that we cannot keep up with naming and describing them. Do you see a shift maybe among your colleagues? Do you think there will be changes in how we look at from a taxonomic perspective? Do you think that we will ever be able to name and describe all the OTUs? And I would like from all of you pragmatic answers.
So the short answer is absolutely not. We're not going to name everything. And the things we name, we oftentimes don't even get to observe again in nature on another recollection because they've already gone extinct, or the habitat, climate change, human perpetuation have already eliminated them. But I do think that taxonomic names and naming of things is going to stay with us. And no, we're not going to fill the taxonomic gap. That's not going to happen. But we're also not going to get rid of the names for the dinosaurs or for the fluffy mammals and the feathered birds. Those names are going to stick. I think what we can do as taxonomists, and the best we can do, is to make sure that the names that do get used are the most accurate for the organism, the specific organism that that DNA sequence comes from, such that it can make an accurate reference. And then everything else that we showed is closely related can be binned and talked about in an intelligent way. I mean, I don't work in the microbial world. A lot of my colleagues are microbiologists. I feel sad for them that they can't name things, but you know, we are fond of names, and that's the human connection. We've been doing it for thousands of years. We've been naming organisms. So I don't think it's going to go away, but no, in the real world, we're not going to be able to do all of them.
Yeah. Do we lose or do we gain things? You know, moving a little bit from species names, this molecular name, which we already have right now.
Well, you can have it both ways. So you obviously lose some value in dealing with names, but in a way, despite the fact that there's sometimes of vital importance to be able to name and identify an organism that you're in interaction with, it's not necessarily in every case when we, for example, talking about practical significance of an identification. My favorite example in this is bird strike identifications, where for people who making decisions to manage wildlife at our airports, they're not necessarily concerned about the species name. They're more concerned about the size of the bird that the plane collides, because it has different risk to the aircraft and then the consequences. So as long as they can get sometimes a genus level identification, they can, and they know information about the habitat that this genus occupies. They can decide how to control the wetlands, for example, around the airports where they collide with geese. So that's one of those examples where it's not always about the species name. But obviously, not to repeat myself, like we are gaining, consider that there's a reinforcement, we actually gaining capacity to accelerate registry of life on the planet. And we can do it with high accuracy and cost. In fact, a lot of the activity that we have been doing at the Center for Genomics, we can call it reverse workflow, because we can quickly and cheaply put those individual specimens to a cluster, give it a name, give it a bin, and then allow the taxonomists to browse through the database, explore the images, request a loan of a voucher, that they focus their effort to describe the new species or catalog. So I see that again. We definitely losing some emotional attachment to those names, because we speak the same language and we understand when we refer to something by a Latin binomial. And then there is some inherent characteristic about that species can be described in that name. And it also points to its place in the tree of life and what role this organism may be playing in the ecosystem. So if you're just calling something by abstract number, it's not easy to connect it to this functional aspect. So those would be probably a few of the examples, but not exhaustive.
So we have been talking a little bit about taxonomy, about names, identifiers, but with this massive amount of data, we need to talk about data, about platforms that can post the data, integrate the data, and Tobias is representing the largest platform for biodiversity information, more than 4 billion records. So how does your platform integrate this wealth of data, taxonomic names, with molecular sequences? You know, thinking about the next 5 years when you will have more data, Sorry, I guess I should, now I cannot repeat everything, but I will just say that I'm inviting Tobias to answer a question about data, storing data, integrating data, connecting DNA sequences with species names, with molecular identifiers, other metadata, and kind of scaling up for what is coming from the eDNA metabarcoding.
Sure. Yes. Yeah, in terms of standards and infrastructures, I think we're actually in a good place. I think with the GenBanks and the biodiversity databases like OPIS and GBIF, ALA, I think it actually serves the purpose and the data standards to accommodate what is needed to mediate the information we have in the DNA is also adequate. And then in GBIF, or that means in Catalog of Life, both the bold bins, the system of identifiers, and that of unite species hypothesis, but also genome taxonomy database is integrated there in the sort of classical taxonomy as unranked children of something, which allows GBIF to index those codes as species in a taxonomic tree, you can say. Of course, there's a big portion of the tree of life that is not covered then, and they can only sort of be referred to the most to the taxon level that we can identify them to, which is often kingdom bacteria, something like that. So we need some systems for the lagging parts of it. I don't know if anybody is going to build that, but it works well for what's there already, I would say. And then of course the linking to INSDC and things like that is important here.
Thank you. I will come back to Tobias and Evgeny, but I'm just going to use the microphone once. How do you think AI is going to help us or not, but mainly help us name these molecular identifiers, these genetic clusters that we have out there? Or do you think it's possible?
It's already being done. I mean, with the BOLD system and the unite species hypothesis, and I think it works really well. It's a big task, and then applying it to all genetic markers and all organism groups is a big mouthful. But it would be great if there would be the capacity and resources to do something like that. But it takes resources. Can AI help? I mean, you could just automatically put Linnaean names on everything we can identify, but that would be really unsexy. I mean, it wouldn't appeal to anybody to just have auto-generated names for everything. I don't think so. And the codes work very well. So I think whenever we find some molecular species that we can link to specimens or an organism, which turns out to be something we want to communicate about, I think we're just going to put good old classical names on them. So, but of course, there's a lack of effort of taxonomists to do that.
Yes, I agree. It's already happening. And in short term, there is a huge advantage of using the tools just to automate some even curatorial activities. For example, at CBG, every specimen that is moving through the analytical workflow, especially those tiny flies, they get imaged, and then we don't even do any morphosorting. The algorithm is actually assigning them to order or sometimes to a family level with surprising accuracy. And that information later down the stream is actually converging with the sequence information and is part of the validation process. So you can identify errors and any potential artifacts. So that's one of the examples. And then subsequent curatorial activities, just trying to see any discordances between metadata that is supplied with the specimen. All of that is part of those examples. But in the long term, you can actually see that as the training data sets are improving, you can move to the next level of aggregating and synthesizing information about morphological characters, any distribution information, and maybe also looking towards formalizing some next version of the automated species description. Whatever they are called, that's a separate meta. And perhaps we'll hear some of that from Rodolphe Meyer's plenary tomorrow.
Thank you so much. Now, we started with taxonomy, then data, infrastructure. But data, it's nice to have. It doesn't mean that it gets translated into decision, right? We saw the plenary this morning. We talked about data moving to information, knowledge. It doesn't mean that that knowledge will be used in decision making. So I have this question for Mylene and Tobias. They are not decision makers, so I cannot ask them what kind of data they would need to make their informed decisions. But they are working with decision makers and they are involved in projects at the science policy interface. So they are familiar with what it is expected from the scientific community that is generating this DNA data. And if you can give me some examples of, how do you use eDNA and eDNA data into conservation, into environmental reporting? Do you think it's accepted by decision makers? Do you think they are still waiting for something from our community? Miley.
Thank you, Adriana. Well, I think that we are all decision makers to a certain extent. And that is an important reflection for us because that means that we look at all the different sectors of the society as a decision maker. So local communities, government, I don't know, ourselves as academics. And in that sense, what we have done so far is try to do capacity building in genetics, concepts, genetic data interpretation for all the different sectors of the society. Because you cannot ask, for instance, the government or the enterprises to adopt genetic tool if they don't really understand what is behind, what is like the limitation, what is really, as you were saying, there are limitations, there are advantages, what is the cost also of doing that. And for instance, well, we have been working a lot with DNA barcoding in the context of illegal wildlife trade. But to be able to do that, we have been training the employees at the rescue centers, the employees at the Ministry of Environment, the people from the police officers, people that will never maybe go back to the lab and do the sequence, but that they really understand the power of that kind of data. So that's the way we have found so far for them to really apply and promote and use this kind of information. For metabarcoding, it has been more challenging. We have an example. I think it was 3-4 years ago. There was a pilot research program in the context of fracking. So there was going to be fracking licensing here in Colombia. And we were able to argue that it was important to generate a baseline of microorganisms from groundwaters, soils, sediments, based on metabarcoding. And we did it. The enterprise did the baseline. We did the baseline of the impact zone. The national authority permit had to engage people, experts on DNA metabarcoding data in order to understand the results that we were delivering. But at that moment, we realized that it was so expensive to do that in Colombia that it was not really responsible to us, the government, and as the enterprise to start delivering that kind of results because that was going to be super expensive. So if you want that to be adopted in policy, in countries like this, you need really to build the capacity in there in such a way that that data is cost effectively generated. That will be my reflection there.
Yes, thank you. I mean, GBIF is often or generally together with OPE is the sort of go-to source for when people need big data sets to do decision making and policies. And so I think in that way, I mean, the DNA data already goes into that. Maybe the decision makers don't know, but I mean, some of the detections they are looking at is from DNA. But if you look at the sort of the broader unnamed taxonomy, eDNA, is that going to be trusted? It'll take some time, but there are initiatives to build ISO standards around some of these methods, and I think we might some researchers might be overly optimistic that if we just generate good evidence of something, then people are gonna base their decisions on that, and I don't think that's how the world functions. But if we need to understand how policy making works. So ISO standards is a good approach to going to that. But there is in New Zealand there is this Tiki index, which is partly based on unnamed sequences of classifying rivers as good or bad, and as far as I know, that's being used. Yeah. As a side note, I can say that I used soil eDNA to help the Swedish police put a guy in jail. You can ask me about that later.
NDA.
Thank you so much. Very, very interesting case studies and very, very good opinions. Taxonomy, data infrastructure, decisions. The future. How do we build capacity and trust at the global level? And I'm using this term of capacity building, although I don't think it's very accurate. I think there is a lot of capacity in the global south. Maybe access to technology might be a bit more limited than in the global north, but there is capacity and there is expertise. And I have to say, just before the world was shutting down in 2020, we were in February 2020, we were in Cali with Mylene, who organized a regional workshop to train people in DNA barcoding in the context of illegal wildlife trade. And it was one of the best workshops that I attended. And I was an instructor, but I was actually learning more than I was teaching. So there is huge capacity. We just need to look for it. So, Mylene, you live and you work in Colombia, one of the mega diverse countries. How do you see this? We call it capacity building for now because I don't have another term. But how do you see this for the global south? You know, knowing that there is a decreasing number of taxonomies, there is an increasing number of DNA data being generated, there is this flood of information. How do people from the global south get trained in understanding the data, analyzing the data, using it in biodiversity conservation and sustainable use?
Explain. Okay. Okay, how we do capacity building in here, or what is like our aim, as I was telling you, for us, something that is super important is to tackle like different sectors of the society. Not only like the future scientists, but that the people that will fund the research, the people that we use the research, that they understand why this is important. So we have been like really making a lot of effort in doing capacity building, not only for future scientists, but for the rest of the society, so they can understand, respect, and use the kind of data that we generate. A limitation that we have, I think, is really like the cost. The cost, because the technology is super expensive. The business model of most of these enterprises, I don't know why they have like a different policy for Latin America. I mean, Oxford Nanopore Technologies, that for a moment it was for us like, okay, this is democratizing the DNA sequences. When you were in Europe and the US, you can buy like a pack of cells that makes that each cell flow is less expensive. In Latin America, you can only buy it by unit. You cannot buy the pack. And so the price for us for doing sequences is at least 40% up to 100% more expensive than doing that in what we call the global north. And so I think that is something that we need to change, because we have this challenge of generating the information. We have the context for using, we have the capacity, we have the researchers, but generating the data is super challenging in terms of cost. And I don't have the solution. I mean, I would like to say that we can put together the Chamber of Commerce and we have like a special rates for biodiversity or things like that. But that is something that we need to keep in mind when we're talking about generating data in this challenging context when it's so expensive compared to other places. That's what
Thank you, Mylene. This is a very important point that Mylene is raising, and working before the pandemic at the Center for Biodiversity Genomics, I had the chance to be involved in various workshops around the world, funded by the government of Japan, through the CBD. And that was always a problem in all the labs that we were hosting these workshops. The cost of reagents in any country in Africa or Latin America or Asia is much higher than the cost of the same reagent in Canada or Europe. So what is the solution? I guess a lot of lobbying at higher levels, at World Trade Organization and so on, because it does definitely impact the research in the Global South. Now, going back to taxonomy. Regina, you've been involved in this field for a very long time. Do you see that there will be trust, or there is less trust between hardcore taxonomists and molecular people and decision makers? Like, can we build trust better between different sectors? And I'm saying that because we expect the taxonomists to do a lot of work for free because they work in museums. So it's their role to identify our samples for free, which is not really the case. So there is maybe a bit of reluctance from that group to embark in this big DNA enterprise, because maybe they don't see themselves, represented or being thanked for their work. Do you see trust being built, being increased? Are you optimistic or not?
Very complicated question, can be answered in many facets. Facet number one, the taxonomists that there's some that will not reconcile. Many of them have already retired and they're not being replaced. We, in my community in Southern California, work extensively with the only taxonomists that are truly employed are actually for wastewater agencies, because we have very strict California wastewater discharge regulations. They are the ones that do the monitoring. It's their voucher specimens that come to the museum. I don't go out to collect anything for taxonomy, for new description. It either comes to me or I have separate grants to do that as a research endeavor, not as a museum staff. So those folks that are adamantly against having any sort of, or think that they will be replaced by genetics, are long gone. Contemporary curators and scientists like myself have long ago embraced both of them. The main concern that I have now, as I'm seeing some of this data churn out, is that we haven't given as much thought to exactly which sequence we're going to use to be the representative for a species. When you name a species, you have, or in my case, I have the luxury, usually have a pool of organisms from one place, from one time, one locality in front of me. I will pick usually the nicest, most adult, depending on the taxon, either the male or the female, and then go through this very regimented description. That holotype, that particular specimen, in the ideal world, would give up a tiny little leg for its full genome. We would do chromosome analysis, we do metagenome, we would do everything with it. And then that specimen is in the repository. When we do DNA barcodes up until now, and I'm now myself coming to realize that this is a real issue, is we apply a name. So if the specimen's type locality happened to be in Vancouver, Canada, and I'm using the same name in Los Angeles or something that looks similar, and it's starting now to go into these bins and into this larger data field, that is the part that I'm like really concerned about. So when we pick up a specimen to DNA barcode or do anything else with, where was it collected? Give me a latitude and longitude. You don't even have to give me a depth because we can calculate that, but give me a date. Because many of the species now, as we are picking them up on our coast, are moving further north as the ocean waters are warming up. So that is where trust of like the names, the few names that we do have that we can apply are applied well and consistently, and then new names get added. And these bins are going to keep changing because so much of it is unknown. We have dozens of undescribed species of isopods in Southern California, right on the beach, where there's millions of people living in one of the largest metropolitan areas. So new species coming up is going to be continuously happening. And the kind of collection methods of where you go out to the shore and you pick up one animal or a cluster of animals is a very different kind of method than when you're going through and using, you know, either soil sample or a liter of water to extract the data. Those are not the same kinds of measures, and policymakers and decision makers have always used. We go out with a net, we count all the fish in the net, we identify all the invertebrates, and then make a decision if the environment has changed or the harbor ocean quality is the same or not. And now we can detect eDNA samples, and it will say that there is a whale in Los Angeles harbor. And it maybe not have been in the harbor, but close by and left DNA. So those two are very different kinds of measures that need to be considered. I know it's a small step, and I now think that probably the taxonomic community is not stepping forward loudly enough to make those baseline things, but contemporary taxonomists or systematists or folks associated with collections and specimens are, you know, completely engaged in this genetic community, and we use those resources well.
Thank you, Regina. Now we have a few minutes to open this for questions from you and for discussion. The whole idea behind this panel was how do we move forward? Well, where we are right now and how do we move forward? We know that if species are not named and described, they do not really exist in the context of IUCN red lists, CITES, the list of invasive and introduced species. So we have this massive amount of molecular OTUs. Some of them will be described, some of them might not be described, but how do we move forward as a community? So if you have comments or questions, please step in. I think we have an extra microphone, maybe? No, okay, I'll pass on this. And then if somebody can look at the Zoom chat, if there are questions on the chat. Anybody wants to start? Okay, she's closer.
Yes, I know it's a tricky question because it's a big discussion in the field of mycology and of botany, where we discussed a lot about which section of DNA will be except for types. It's a very big thing. And I'm wondering if you guys believe that the code is ever going to adopt this in any form, or if you guys have any insight in this.
So, Tobias?
Yes, I think it will. Soon. Yeah. No, but I mean, if you have the option of making a full genome, then you should of course do that. But the classical markers, right? But then I mean, if there ever is a doubt, you can refer to the more full information of the type specimen. Yeah, I'm optimistic with that. Yeah. It's fully needed. There are many organism groups where you cannot use morphology. If you sequence the specimen, you don't have a specimen anymore, right? Unicellular things.
Hi, I have a concern. It's about, in the same way, I'm doing metabarcoding of stomach content of fish. And one of my concerns is the lack of names. Is in the same way that we are moving forward with genetics and barcodes and genomics, in the same level for answering questions of decision making about policies or something in the way of functional traits and ecological problems required to identify the organism. I can find with a one barcode and a specimen, but I can't put it in the trophic change, or in the trophic change, I can put it in interaction with other organisms. For example, maybe invasive species or relationship of some very structural species in the environment. So that might concern how to avoid that, because if we just have a lot of, I have this problem in this moment with my data. I need to understand the trophic web and do some guessing about that. But I just have barcodes, and I cannot infer. I could be irresponsible to infer relationship and interactions to make some decisions in the area. How so? It's a concern. I know. I don't know how to plan a question about that, but I don't know what could be a way to work on that.
I think you have the stomal contact of your fish, right? And you have a barcode. That barcode maybe won't have like a species name because it is not yet like in the DNA reference library, but probably will be already good enough to have a family and maybe a genus level assignment. And I think that then you, so far, while you enrich the DNA reference library of your lake, but I mean, that's important. You know, that's why we cannot forget about the pace of all the other components of that project. But what we do so far is that you take the list of species that are present, like with classic inventories already in the place you work, and you try just to cross-ref so far. But I think that that's why it is so important also to continue working in the enrichment of the reference libraries and the taxonomy if you want to make more sense of your data. But that genetic unit is already important and, let's say, good enough for ecological comparisons, no?
Yes, thank you. My name is Urmas. I'm taxonomist. So, and I think it's in such discussions, it's very important to keep apart two very different things. One is taxonomy, where we basically mostly delimit species. Of course, we are making classifications, identification keys, etc. And then there is classical communication part, which is mostly codes, which actually have regulations how we communicate taxa. So, and now we have to understand if we move some parts of taxonomy into these codes, like sequences, physiology, biochemistry, because there are species which cannot be identified based on sequences, but on physiology, biochemistry, yes. And even we have regulations for those features. So we have to understand that codes are about communication. And as Tobias said already, about sequences, we can already communicate them, at least in GBIF. And when new sequences are coming in, it's very easy to communicate them. Yeah, I will stop here, but I think it's very important to keep apart those two things. And I recommend not to include criteria, traits, or characters, how you call, into this communication part, because this will harm actually this naming system.
Even with those sequences that do not carry names, if there's enough of that sequence in a standardized database like OBIS or GBIF, you can actually infer something about your sequence, right? Where has it been found before? Is it only in pure water? Is it in dirty water? Is it something like that? So we can actually infer quite a lot if we have enough data, right? Yeah.
Thank you. We have We have 5 minutes left, and I'm sure this is a very interesting conversation, so we can continue it after. Now I would like to ask the panelists for a last closing remark, quick, one sentence. Where are we going? Of course, this panel is focusing on taxonomy and DNA technologies for biodiversity inventories. But as we saw at this conference, there are other methods that are being used in inventorying biodiversity, acoustics and camera trapping and remote sensing and so on. But this particular panel is on DNA. So remarks on the future of biodiversity inventories for action, basically, for decision making.
I think maybe I'll draw on Malin's comments. I think she and I share a fair bit. It is about communicating to the larger public. I mean, it's biodiversity. We are part of the biodiversity. The biodiversity world is around us. So it has to have a human component to it and a human interaction, because otherwise we're not saving it. And just to have A's, G's, T's, and C's may not be the only value.
I'll make 2 comments. I'll actually make a call to stop buying into hype and chasing the new technology as a single solution to all the problems and throwing baby out with the water, leaving behind all the good knowledge that has been gathered so far. And I would also make a call for collaboration rather than competition. It's very easy to criticize all the big initiatives that are trying to produce large volumes of data, and they always come with certain errors in them. I would ask everyone who is trying to explore this data and identifying errors, ask yourselves like what you have done as a community to contribute to address those problems and help those big databases, whether it's BOL, GBIF, to identify and correct those records. So then it will be much more fruitful for getting us high quality reference libraries for any application in the future.
I very much agree with the 2 before. I can add that there's a very high hope for AI to solve some things now, and I think the two of our keynote speakers, Sabine and sorry, what's her name? Alice. Yeah. So, it was amazing. I mean, how they really pointed out that we need to keep as you say, the human in the center of things, and even if we can build absolutely amazing models, we still lack a lot of data for those models to be performant. So we should spend them to inform us on how to mobilize existing data and where we should generate new data and things like that, and help us with the boring steps, but we should keep the human in the middle of everything.
Well, I agree with the 3 of them, but no, but I think that in 50 years, most of the inventories will be done with passive sampling, camera traps, sounds, eDNA, and I think that what our job for like calibrating those models or generating the data is what we're going to do in the next 50 years. But after that, I believe that will change a lot. I mean, I imagine that a species will be identified maybe in clusters, but based on images and based on all the information that we have built from DNA and from taxonomy. But I do believe that will be completely different from now, even if we should not forget the human component of it.
Oh, yeah.
Thank you so much to all of our panelists. I have to say I'm happy that I didn't hear from any of them saying that we will go species free and we will just have 0 and 1s and so on. I think we are still very much connected to names. For some reason, we are humans. We like names. How do we make order in all these names? How do we advance with naming what is unnamed? I think we don't have an answer for that. I think the future is quite exciting. I know we rely a lot on AI, but yeah, I think AI cannot solve everything. And this is another call, I think, for more collaboration. I know I sound like a broken record, probably, that we need to collaborate. But we are gathering data so fast, and it is so easy to lose track of what we are trying to do, actually, besides generating data. How are we going to use that? How are we going to conserve biodiversity and use it sustainably if we focus on just generating data and more data and more data? So if you have other comments, I mean, we are here the whole week. We will be downstairs in the poster session. If you have case studies on how you are using DNA data for your own project or for your own collaborations with decision makers, especially so we can understand what their needs are, please come and talk to any of us. But I thank you very much for being here. At the end of the day, after watching slides for so many hours and being indoors. So thank you so much. Oh, one last thing that I forgot to say, and Evgeny also forgot to say. Next year, the beginning of November 2026, there is the next International Barcode of Life Conference in Bangkok, Thailand. Keep an eye on the LinkedIn page and social media, and please come and present your work on DNA and taxonomy and ecology and so on. Thank you. Yeah.